Welcome to our live class for today's lesson. Welcome to our live class for today's lesson. So if you can hear me, you can just give a thumb up or like so that I know the people I am with. Okay, so we are doing neuromuscular junction. So what I just want you to understand, this neuromuscular junction, you help you to understand the mechanism of muscle contraction. Okay, so this neuromuscular junction has events, of which I will show you the most four important steps of event at neuromuscular junction. Now, what is this neuromuscular junction? So this neuromuscular junction is just a junction between the terminal branch of the nerve fiber and the muscle fiber. Okay, just the junction between the terminal branch of the nerve fiber and the muscle fiber. All right. Now the terminal branch of the nerve fiber is what we are calling the axon. So this is the axon here. This is the axon. So this is the axon. So the axon is the terminal branch of the nerve fiber. The terminal part of the nerve fiber is what we are calling the axon. Okay. Now this thing you are seeing like a bulb here on the below the terminal nerve of the nerve fiber. This thing you are seeing like a bulb here is called the motor end plate. So this is called the motor end plate. Motor end plate. Okay. So this is called the motor end plate. Okay. So now this axon terminal contains what we call the mitochondria and the synaptic vesicles. So you know from the cell biology or the cell physiology to say the mitochondria is the powerhouse, or something like that. So it has ATP like that. So this axon terminal, it has mitochondria and synaptic vesicles. So this structure seen here on the motor end plate, which is the bulb thing like. So this thing with inside, which is folded, this is what we're calling the mitochondria. This is the mitochondria. Okay. This is the mitochondria. Okay. And this vesicle we are seeing here with dots inside, this is what we are calling the synaptic vesicle. So this is the synaptic vesicle. Okay. Now, this synaptic vesicle contains the neurotransmitter substance. This one, these dots we are seeing inside, they are not just dots. These are neurotransmitter. So this neurotransmitter is what we are calling the acetylcholine. Acetylcholine. I'll show you how the acetylcholine is synthesized. Okay. So this acetylcholine is basically synthesized by the mitochondria, which will be present at the axon terminal. So it's going to be synthesized or made by the mitochondria present at the axon terminal and stored in the vesicles. So when they are made by this acetylcholine is just a mixture from since we know the mitochondria is involved in Krebs cycle. From there it will mix with the choline. The choline is what we are obtaining from diet or from food. So this reaction will combine with the choline, which is from the mitochondria, and together they will fuse and they will form this acetylcholine, of which I will show you like a simple equation so that you may understand. So this mitochondria, it contains ATP, like I said, which is the source energy for the synthesis of what? This acetylcholine. Okay. For those of you who are joining, welcome to the live class. Feel free, if the concept seems to be unfamiliar to you, you can ask so that you are together, right? Okay, so you're supposed to know where the presynaptic membrane is and where the postsynaptic membrane is. Okay, so now the membrane of the nerve ending is what we are calling the Pre-synaptic. The pre, of course, from the prefix, you can say the pre is before. Okay, so the membrane of the nerve ending is a presynaptic membrane, which is before the synaptic membrane. Okay, so the presynaptic membrane is this part here, before the synaptic cleft. So this is the presynaptic membrane. Presynaptic membrane. So this is the presynaptic membrane. Okay. Now, have you seen the space here? There's a space between this bulb. You're seeing here this bulb and these folded neural folds. I don't know, subneural folds. These subneural folds and this bulb. There's a space here, and this space is what we are calling. This space is called the synaptic cleft. Synaptic cleft. Okay. This space is what we are calling the synaptic cleft. So the synaptic cleft, how can you define it? So the synaptic cleft is just it's basically the space between the two membrane. Which membrane? Of the postsynaptic membrane and the presynaptic membrane. So meaning the postsynaptic membrane is this part here. After the synaptic cleft, or the space between, the space in between, this part here below is what we are calling the postsynaptic membrane. You're supposed to know this. The postsynaptic membrane. Okay. Now, this the same space which we are calling the synaptic cleft. This it contains the basal lamina. So it is a thin layer. This basal lamina, it is a thin layer of spongy reticular matrix through which the extracellular fluid diffuses. Okay, so an enzyme, there is an enzyme which we are calling the acetylcholine esterase, or abbreviated to be ACHE, acetylcholine esterase. Let me just write the full name of the enzyme: acetylcholine esterase, like that. So this enzyme is attached to the matrix of the basal lamina in large quantities. Okay. So yeah, like I mentioned, you have seen these folds. We know now the cleft. We know the vesicles, synaptic vesicle, which contains the neurotransmitter. We also know the mitochondria, which is the powerhouse, which performs the important function in formation of acetylcholine. Also know the presynaptic membrane. We know the axon. Okay, right. Now, what about these folds? So these folds, already I mentioned them to say these are subneural clefts. So these are subneural clefts. Okay, these are subneural clefts. So these are subneural clefts. So these subneural clefts, they are at the postsynaptic membrane. So this postsynaptic membrane is just the membrane, the mass of fiber, and it is thrown into neurons of folds. And these neurons of fold are the same one we are calling the subneural clefts. Okay, then on the postsynaptic membrane, we have the receptors. On the postsynaptic membrane, we have the receptors. When actually the acetylcholine is released from the presynaptic membrane on the motor end plate of the bulb, yeah, they'll actually bind to the certain receptors on the postsynaptic membrane, and these receptors, we are calling them the nicotinic acetylcholine receptors. Nicotinic acetylcholine receptors. Okay. Now let me talk about the neuromuscular transmission. Then I give you the events that occur at neuromuscular transmission. Okay, so neuromuscular transmission. So neuromuscular transmission, this is just the transfer of information from motor nerve ending. Now, by this time around, when I mentioned the motor nerve ending, you bear in the mind. I'm sure you know in the mind by now. So this is the transfer of information from motor nerve ending to the muscle fiber through the neuromuscular junction. Now you now know the neuromuscular junction. So it is the mechanism by which the motor nerve impulses, they will initiate muscle contraction, of which will be our next episode. When you understand the neuromuscular junction and the transmission, then the muscle contraction will become very easy for you. Okay, now let me run through the events that occur in neuromuscular transmission. So events at neuromuscular transmission. Okay, event of neuromuscular transmission. Okay, what are these events? Number one, at neuromuscular transmission, there will be release of acetylcholine. Okay, two, there will be action of acetylcholine. There will be release of acetylcholine. Two, there will be action of acetylcholine. There will be action of acetylcholine. Then three, there will be development of end plate potential. Development of end plate potential. Okay, then the last one, for which will be, there will be destruction of acetylcholine. There will be destruction of acetylcholine. Okay, right. Okay, I hope you guys were with me. Okay. Right. Those who are joining can join. Okay. I want to show you something. Right. Okay, so this is how the transmission is moving, how the information is moving. So the information is coming from the spinal cord. Okay, so what you are seeing here is just the section, cross section of the spinal cord, which is just been sliced. Then this is the section of the spinal cord, the cross section of spine. So this part here is the posterior part of posterior side. This is the posterior side. When I say posterior, I mean back side. Then this is the anterior, which is the front. Okay. Now the information from here. Okay. Okay, like when you touch in an instance where you touch the hot object, the way your hand reflexes. So this is how the information is being transmitted. Okay, so that you move the hand and hence causing the muscle to contract or relax, something like that. So this is how the information is being moved. From here, the information will pass through this, what you are seeing here. So what you are seeing here is the motor neuron. Okay, the motor neuron, which has the myelin sheath. This thing you are seeing, these are the myelin sheath. Of course, if you watch the video, you understand the neuron, the structure of the neuron on muscle neuron neuron tissue, okay, on nervous tissue, actually, on in anatomy video, the previous anatomy video. So if you haven't watched that video, it's supposed to go. Like some of the things I won't be repeating, okay. It will be based on those foundation. Now, so this you are seeing here, neuron, basically just the, specifically I can say it's the alpha motor neuron. This is the alpha motor neuron, okay. This will be the alpha motor neuron, and these channels you are seeing here, okay. These channels you are seeing here, these channels are sodium sensitive channels. These are sodium sensitive channels, right? So the structure seen here with folded structure inside is what we are calling the mitochondria, which performs actually the most important function in synthesis of what, the acetylcholine. Yeah, and this channel is the one which transmits the choline from the diet, okay. So this is the channel which transports the choline from the diet, to make what, in the acetylcholine, okay. And this thing you are seeing with dots inside, it's not a ball actually, no. Yeah, so this thing you are seeing here, it's a synaptic vesicle which contains the neurotransmitter inside, and this neurotransmitter is called the acetylcholine. So this is the synaptic vesicle. So this is a synaptic vesicle which contains the neurotransmitter, the acetylcholine. This is the abbreviation, okay, the acetylcholine. And this structure seen here is just the zoom part of this synaptic vesicle, of which I will show you how the acetylcholine moves and fuses with the presynaptic membrane and enters the synaptic cleft until it reaches the postsynaptic membrane, okay. So, and these channels you are seeing here, these are what? These are voltage sensitive calcium channels. These are voltage sensitive calcium channels, okay, right. These are voltage sensitive calcium channels, okay. So the information is coming from the spinal cord. Like I said, this is just the striatum of the spinal cord, which has the posterior and the anterior part. And then when you touch the hot object, the way you reflex the hand, like I said, you contract. It's either your hand you contract or relax or something like that. So the information is coming from this side, passes through the alpha motor neuron, and it reaches this motor hand plate, which is the bulb. And this bulb, this terminal has certain channels. There's actually the synaptic vesicle, which has the acetylcholine, the mitochondria, which is the major part low that produces what, ATP. It is also involved in what, Krebs cycle, all that, until the formation of acetylcholine. Okay, now what happens at neuromuscular junctions? What happens? So I've given you the events to say the four events. The four events, number one, I say there will be release of acetylcholine. Two, there will be action of, when acetylcholine is being released, it's supposed to act its function. So there will be action of that choline, acetylcholine. Then from there will be development of handplate potential. When it develops, there will be now destruction of acetylcholine. Since it has finished its function, it's supposed to be distracted, and it's within millisecond it's distracted. Okay. Now, when action potential reaches the axon terminal, so when the action potential actually, so in the action potential here, right? So this is the action potential from the spine. Okay, it's moving, huh? So these are the myelin sheath. You know the function of the myelin sheath. You need to watch the previous session on nervous tissue anatomy so that you may understand. So when action potential reaches the axon terminal, it opens the voltage gated calcium channels membrane. So now, before it opens these channels, okay, there will be influx of sodium. Okay. There will be influx of sodium You know, sodium it's a positive charged cation. So when there's influx of these cations, will cause the terminal part inside the terminal to be what? Highly positive charge, eh? Okay. So there will be more of the positive inside the terminal. Okay. And this positivity will cause the opening of these voltage sensitive calcium channels. Will cause the opening of these voltage sensitive calcium channels. Okay, so like I said, let me repeat the pointer to say when the action potential reaches the axon terminal, it opens the voltage gated calcium channels, the membrane of axon terminal. Now, when these calcium ions from extracellular fluid enters the axon terminal, okay, when this enters the axon terminal, so this when the charge inside will cause the opening of these voltage sensitive calcium channels. So there will be influx of calcium here, influx of calcium. So calcium enter inside the terminal or the mount plate of this bulb. Okay, and this will cause the bursting of when the calcium ions from extracellular fluid enters the axon terminal. This will cause the bursting of vesicles by forcing the synaptic vesicles to move and fuse with what? The presynaptic membrane. So this membrane you are seeing here is the presynaptic membrane. Alright. Now the acetylcholine is released from the ruptured vesicle gate by what? It's going to be released by exocytosis. And the acetylcholine will diffuse through the presynaptic membrane and enter the synaptic cleft. Like I mentioned, the synaptic cleft is just the space between the presynaptic membrane and the postsynaptic membrane. Get? Yeah. And in between those space or on the postsynaptic membrane, there are receptors there, which are called the nicotinic receptors. Okay. Now, the acetylcholine is supposed to perform its function, right? Now, before I go on the action of acetylcholine, I want you to show you something very important. Okay, so on the synthesis part of acetylcholine, so we have the mitochondria, okay, and the choline from the diet. Okay, these two. So from the mitochondria, I know this performs a major role in Krebs cycle, where there will be formation of acetyl-CoA. So this there will be acetyl-CoA, and this is choline from the diet. And then when these two fuse, these two reaction, so there will be a reaction, and this reaction they will cause the release of CoA. So the CoA from the acetyl-CoA will be removed in this reaction. The CoA will be removed. When the CoA is removed, what are we forming? We're forming what? The acetylcholine. Now, what's the enzyme which is stimulating this reaction? The enzyme which is stimulating this reaction, the enzyme is called what? The enzyme is the choline acetyl transferase. The enzyme is called the choline acetyl transferase. Okay, well, this was just the side learning so that you know how the acetylcholine is formed. Okay. Okay. So in this vesicle as seen here, there will be acetylcholine. Okay. There will be acetylcholine. Okay. There will be acetylcholine. Now, I've already explained how this acetylcholine is going to be moved from this terminal into the synaptic cleft. Now there are very important proteins that you're supposed to know, and these proteins are what we are calling the snares. Need to know, very important. So the snares, okay, the snares, okay. If the concept seems to be unfamiliar, you are free to comment or give a thumb so that you will be with us. Alright. Okay. Before the acetylcholine, these molecules, before they are being released from here, actually there are proteins which are acting a major role or playing a major part on this release of acetylcholine. So this protein you are seeing here, the greenish protein here, this we are calling, this protein name is called the SNAP25. This is SNAP25. This is SNAP25, and this the other part on the right side, this protein is called the syntaxin. Okay, this is called the syntaxin protein. On the syntaxin protein, there's another protein you are seeing here, which is the orange one on top. On the synaptic vesicle membrane, it is on the synaptic vesicle membrane. These proteins are attached on the synaptic vesicle membrane. So these proteins are supposed to intertwine or fuse so that they will bring this vesicle on the surface of the presynaptic membrane so that it diffuses. Now this protein is the orangish one is called. This is called the synaptobrevin. This is called the synaptobrevin. Synaptobrevin. Okay. Now, what about this one on the left part? On the left part, this orange protein is also called, there's a name. These proteins are very important, supposed to know. So on this protein is called the synaptotagin. Synaptotagin. Okay. This is called synaptotagin. So these one, the orange one, they are bound to what? The synaptic vesicle, which contains the acetylcholine. Okay, like I said, these proteins, they are called the snares. The name of the protein, we are calling them the snares. These are the snares. So we have actually two. We have the V snare and the T snare. So we have the V snares and the T snares. Okay, so now on the V snare, what protein do we have? On the V snare, the V means the vesicle. Okay, the V means the vesicle, and the T means on the transmembrane or the membrane. Okay, so it will be easy for you to classify these proteins. So the V snare, these are the proteins that are found on the synaptic vesicle. So on the V snare, we have the synaptobrevin. And another one is called the synaptotagin. On the T snare, we have the SNAP25. Okay. SNAP25, right. Another one, we have the syntaxin. These, they are very nice to sound. They sound like names, eh? So these are, we have the SNAP25, we have the syntaxin. Okay. Okay. Right. Okay, now when these snares intertwine, when these snares or proteins wrap around each other or intertwine, they will pull. So what will be happening here is that they will be intertwining each other. The SNAP25 and this synaptotagin, they will intertwine, and even this one they will intertwine. So when they intertwine, they will start pulling this vesicle toward the presynaptic membrane. Okay, when the protein snares, they will pull the vesicle so that it diffuses with the presynaptic membrane. Now, do you know the role of the calcium? The role of the calcium, which is coming from the extracellular fluid. So the role of the calcium it's the one which activates these proteins to activate the snares so that they will start rubbing around each other, or they will start intertwining. So we're supposed to know the law of the calcium. So like in your explanation, they say they may the neuromuscular junction, transmission, explain whatever. You're supposed to know, you're supposed to mention these proteins, the way they're acting, even the law of the calcium. So the law of the calcium, which is actually the influx of calcium from these calcium sensitive calcium channels. The law of the calcium is to activate these snares. When they're activated, they will start intertwining each other and putting this vesicle toward the presynaptic membrane. Okay. Now, when it's being put the presynaptic membrane, what will happen? Through the excitosis, this vesicle, this membrane will be digested so that it releases the acetylcholine. So now there will be release of acetylcholine. Okay. Wow. So now when the acetylcholine is being released, where does it go? When the acetylcholine is released. Okay. Initially on the video I said this, I gave you this structure to say. So no, this structure. Actually, this is the bulb, the motor end plate. So this space here between the presynaptic membrane and the post one, and this space is what I call the synaptic cleft. So this is where the acetylcholine will be released. So the acetylcholine will be in this space. Okay. So in this space, will be in this space. I hope you get. Right. So now, what will be the action? Now we are back to the events of the neuromuscular transmission. What will be the action of acetylcholine? What will be the action of the acetylcholine? Now after entering the synaptic cleft, the acetylcholine molecules, they will bind to what we call, according them, they will bind to the receptors, what we are calling the nicotinic receptors. And these receptors, they are found in the postsynaptic membrane, and they will form acetylcholine receptor complex. Okay, and it will increase the permeability of postsynaptic membrane for sodium by opening the ligand-gated sodium channels. Now, sodium ions from ECF, the extracellular fluid, they will enter the neuromuscular junction through these channels. Okay, let me show you what I'm talking about. Let me remove my snares here. Okay. Right. Okay. So now, okay, the space which is on the superior part here, there's a space actually. So this space, that's where the acetylcholine is. Okay, which is the synaptic cleft. Okay. So after entering, when the acetylcholine reaches the synaptic cleft, they will bind. So this receptor here, this thing you are seeing like a cup here. It seems like a cup. It's not a cup, right? So this part, which is you are seeing like it's a cup and there's like a hand. So it's a cup and having like a hand. Okay, so the thing you're seeing like it's a hand here. It's not like a hand. It's a receptor. We're calling the nicotinic receptor. So on these receptors, or this question, I've seen a lot of questions on this neuromuscular junction, more especially on these nicotinic receptors. They ask you on MCQ questions. Okay, right. So this acetylcholine, it will bind to this. So this acetylcholine, when it's being released, it will bind to this receptor. What happens when this molecule binds to this receptor? So this receptor, I said, is present in the postsynaptic membrane. So when it binds there, to form the complex. The complex is called acetylcholine receptor complex. When it binds there, have you seen on this same channel? This channel is called the ligand-gated ion channel. This channel is the ligand-gated ion channel. It's a ligand-gated ion channel. So this ligand-gated ion channel. So if you look inside, it's like it's having a gate. So that's why you're saying ligand gated. It's gated. It's having like a gate. So when this acetylcholine bind to this receptor, there will be opening of this gate inside. You're seeing this gate inside, it will open. And this fold you're seeing here, the folds here are seen is the functional, the post junction folds, post junction folds, or the subneural clefts, okay, right. So when this binds, the opening of ligand gated ion channel. When this ligand gated ion channel, what happens? When this channel opens, now the sodium, sodium, we know already to say sodium on the ACF, extracellular fluid, sodium is high in concentration. Okay, so when this opens, there will be influx of sodium. Okay, there will be influx of sodium. Sodium will start entering the postsynaptic membrane, start entering via the ligand gated ion channel. So there will be sodium inside here. So sodium will be coming inside. Okay. So there will be more, since there's more of sodium outside. So the opening of this, there will be more influx of sodium. So sodium will enter the permeability, since the permeability increases. So meaning we are making the inside of the cell to become more positive. Okay, there will be more positive inside the cell. There will be more positive, more positivity. Now this positivity, this sodium, they will change, or they will alter the resting membrane potential. And this resting membrane potential, when it alters, mostly the resting membrane potential is actually somewhere negative 90 millivolts. So now when this sodium alters the resting membrane potential, what does it develop? It's going to develop the electrical potential, which you are calling what? The handy plate potential, sorry. The handy plate potential. So this would develop what you're calling the end plate potential. I repeat, binding of this opens the ion gate. When the ion gate opens, there will be influx of sodium. The more influx, since there is more of permeability, meaning from the ECF, the sodium will be entering, and will form the end plate potential. Okay, there will be influx of sodium. Okay, now we are on what event? We are now on event 3, which is the development of the end plate potential. There will be development of end plate potential. Okay, development of end plate potential. So the end plate potential is just the change. Okay, when this develops, this end plate potential is just the change in resting membrane potential when an impulse reaches the neuromuscular junction. When an impulse reaches the neuromuscular junction. So like I said, resting membrane potential at neuromuscular junction is somewhere negative 90 millivolts, negative 90 millivolts. Okay. So when sodium ions enter inside, there will be slight depolarization. Okay. There will be slight depolarization. And this slight depolarization occurs up to what? Negative 60 millivolts, which is called the end plate potential. Slight depolarization. So let's remember potential is this one. So when there's influx of sodium, since the permeability increases, there will be upshoot up to somewhere negative 60 millivolts. Since the cell inside becomes more over the positive gate. I hope this makes sense. So there will be slight depolarization. Okay. Okay, let me show you from the sketch. This is more of the skeletal muscle. Okay, so let me show you this graph. Okay, so this is the resting membrane potential. This is the threshold potential. Okay, this is the checklist page. All right. Okay, so now when there is influx of sodium, more permeability of sodium inside, there will be change of resting membrane potential to this memory potential and form the end plate potential. So meaning there will be slight depolarization. This slight depolarization, let me use a different color. So there will be slight depolarization, meaning the graph will start looking like this, going up, slight depolarization. Okay. Okay, well, the slow connection. Welcome back. So this slight depolarization is what we're calling the end plate potential. Okay, end plate potential. All right, end plate potential. End plate potential. Okay. All right. I hope this is making sense. I hope this making. Whereas we are remaining with one stage. We are remaining with one stage of neurotransmission. Okay. With one stage of neurotransmission. Actually, there's another channel that I want to show you. Another channel that I want to show you. Okay, another channel I want to show you. Okay, there's this another channel. So this channel I was seeing here is called the voltage sensitive gate sodium channel. This is the voltage. So this is the voltage sensitive or gate. You can say sensitive or gate sodium channels. So what happens is that this channel, it has two parts. It has the inactivation gate and the activation gate. So they got a short part you are seeing here, like a short leg. You can say this short leg you are seeing here. Actually, it's not a leg. They will mark you long. Don't light a leg. It's the activation gate. So this is the activation gate. Activation gate. Right. So and this is the inactivation gate. I'll show you how this gate functions. This is the inactivation gate. Inactivation gate. Okay. Now, at certain voltage, so at a certain voltage, which is negative 55 millivolts, there will be stimulation of this activation gate. When this activation gate opens, there will be now more influx of sodium. Now, we have already developed the end plate potential, which is the slight depolarization, which is helping us to reach the threshold potential. Okay. So this is the end plate potential. This is the threshold. Now we are at this part, negative 55 millivolts. Okay. So when at negative 55 millivolts, there will be stimulation of this gate, and when it is stimulated, it will open and cause the influx of sodium. And this influx of sodium causes more of the inside to be more positive. So when it becomes more positive, there will be what? Depolarization. Okay, depolarization. Okay, the cell, meaning the cell will become more of the positive. Okay, so meaning on the curve, or on the let me show you on the curve. Okay, when we shoot the threshold potential, which is the negative 55 millivolts, so now the graph will start moving since the cell is becoming more positive until we reach the peak. Okay, until we reach the peak there. Okay, have you seen? That's how the graph is moving. So at this stage, it's depolarization. This stage is what? Depolarization. Okay, which will be the activation gate will open. When it opens, there will be influx of sodium, and this influx of sodium causes depolarization, meaning the more positivity of the cell inside. Okay. Now what happens with this gate? Now I've shown you the function of the activation gate. Now what's the function of this gate, inactivation gate? This inactivation gate here, what is the function? This inactivation gate is the one which blocks the sodium channel when we reach when we reach positive 30 millivolts, so that sodium influx can't occur. There will be no sodium influx at that stage. So at this stage there is no sodium influx at positive 30 millivolts. Here at positive 30 millivolts, there is no sodium influx. Okay, now from there what happens? Since there is no sodium influx from there there is no sodium influx. What will happen next? Okay, this is just the continuation parts. Just carrying on the same page. Now since we have reached the positive 30 millivolts, there are this channel, and the channel name is called potassium leak channel or potassium channels. Of these channels. So what's the function of these channels? Since inside the cell is now more positive, you get, so the inside of the cell is now more positive, so there will be, since we know the concept to say from the actually the beginning of the physiology on the fluid body compartment, you learn to say that sodium outside is more and potassium inside is more, get. So potassium inside is more and the outside is less, and sodium outside is more and inside the cell is less. So meaning here, this one, there will be efflux. This is not influx but efflux. There will be efflux. So we are actually, potassium will be leaving the cell. Okay, potassium will be leaving the cell. So there will be, so since potassium inside, concentration of potassium inside is big or great, so than outside. This is the concept I was saying. Okay, so this is happening at positive 30 millivolts. Okay, so when we know that the potassium is a positive cation. When start leaving the cell, what happens to the cell? The cell starts becoming what? Negative. That is, so this stage we are calling the repolarization stage. Not depolarization, but repolarization. Okay, so this is the repolarization stage. Okay, this is the depolarization stage. Okay, let me just show you. Okay, this is the depolarization. Okay, so now, so the graph, when cations start leaving the cell, the graph will start dropping like this until it reaches the resting membrane potential. This is the resting membrane potential. What is the resting membrane potential? It's negative 90 millivolts. So this stage is depolarization. That is the efflux of potassium cations or ions, to say. Okay, now, so when we reach the resting membrane potential, what happens to the channel of the channel of the potassium? So we know the channel of potassium is like this. Okay, let me draw. It's like this. It has two gates. We have the inactivation gate and the small leg here, which is the activation gate. This is the inactivation gate. Okay, now potassium is leaving. Okay, now what happens when we reach positive 30 millivolts? This gate will close. When we reach the resting membrane potential, this is at resting membrane potential. The cell is back to resting membrane potential. That's why I'm saying the depression or the destruction of acetylcholine is very important, so that you avoid the continuous contraction of the muscles you get. So this this inactivation gate will close to block this, so that there will be no efflux of potassium. So no wonder the cell is at resting. I think this is clear now. Okay. Alright. And there's another structure which you need to be familiar with. This structure. Actually, I will show you this structure in when we start muscle physiology on skeletal muscle contraction and cardiac muscle contraction. But you won't find this to say in smooth muscle contraction. Yeah, so like I'll show you how the structure of the sarcomere function in skeletal muscle contraction. Yeah, and also in cardiac muscle contraction. So you're supposed to go back and watch the muscle tissue video so that you know the structure of the sarcomere. Very important, you get? Very, very important. You're supposed to know the function. Yeah, there's even the idea of the striding filament. I gave you the idea of striding filament. I also gave all the stages of cross bridging. There are actually 5 of them. Yeah. Also know those proteins in the sarcomere. They can come in the MCQ. The nebuline, the alpha-actinin, the titin, the malmacin, which forms the midline there. We have the nebuline, which connects the thin filament to the Z disc. Those beautiful things. Supposed to know them. Yeah, very important. So, actually, though, okay, the thing you are seeing here, this is the sarcoplasmic reticulum. So this is the cell which inside the sarcoplasmic reticulum. That's why we find the calcium ions. Okay. Yeah. So this is the this we're seeing this fodate or structure you're seeing here, the endoplasmic reticulum. Okay. Yeah, and let me just write endoplasmic reticulum. Okay. Yeah, and this you're seeing here is the transverse tubule. This gamma thing you're seeing here is the transverse tubule or T tubule. In instances, they will just abbreviate the T tubule. Yeah. So, and this gate you're seeing here, I hope you're seeing something here. Right. So, this thing here, which is attaching on the transverse tubule, this thing here, which is attaching to the transverse tubule, is called it's a receptor. It is called the dihydropyridine receptor. And the thing which is attaching to the endoplasmic reticulum or sarcoplasmic reticulum, sorry, this is sarcoplasmic reticulum, sarcoplasmic reticulum. So this is called the lionodine receptor, type 1 specifically. So the lionodine receptor type 1. Okay, so all this structure, the simple name we can call it is the triad. Okay, actually, just to summarize, maybe, okay. Like I said, on the motor nerve fiber, the action of potential, there will be now opening of voltage gated channel. Okay, let me just summarize from this part. So from this section, cross section of the spine of the action potential there, when it reaches the terminal, there will be opening of voltage gated calcium channels. And this, when this channel is open, there will be entry of calcium ion or the influx of calcium ion from the extracellular fluid. Then from there will be opening of vesicles and release of acetylcholine. So before this vesicles open, there are certain proteins that's supposed to know. I said the snares. We have the V snare, those that attach to the synaptic vesicle, and the T snare on the presynaptic membrane. This is the presynaptic membrane. From there will be opening of vesicles and release of acetylcholine. So those snares are supposed to intertwine or to wrap around each other and pull the vesicle toward the membrane and cause the exocytosis, so that the acetylcholine is released. So when the acetylcholine is released, it will go to the synaptic cleft. On the synaptic cleft there, it will bind to the postsynaptic membrane. There is a receptor, what we are calling the nicotinic receptors. And those receptors, when it binds there, there will be opening of ion-gated channel. When the ion-gated channel, we know that outside the cell, the sodium is more than inside, so they will cause to increase the permeability, so that there will be more influx of sodium inside the cell, so that we form the end plate potential. So this end plate potential will cause the slight depolarization. Okay, this slight depolarization, actually, there is another channel. From there will be another channel, which are calling the sodium sensitive channel, which it has two gates. It has the inactivation gate and the activation gate. I say the activation gate, activation gate is the small part or the small leg, and then the inactivation is the long one. So when that gate opens, there will be more influx of sodium, so that it reaches the threshold potential, which is the negative 55 millivolts. From there to cause depolarization, and depolarization meaning more inside the cell, they will become more positive. Okay, until we reach the peak, and the peak is somewhere positive 30 millivolts there, and from there there's other channels. Yes, on the channel which we call the sodium sensitive channels. When it's reached the threshold, the inactivation gets to broke, so that there will be no influx of sodium. Now from the peak there will be depolarization, the efflux of sodium, until it reaches the resting membrane potential. Yeah. So this is very, very important concept for you to understand the contraction of muscles. So join me in the next episode as I learn through the muscle contraction. Okay, and thank you very much for those of you who have persevered to the end of the session. Have a blessed evening, and don't forget to comment if the concept seems to be unfamiliar, so that we work together. Okay, have a blessed evening.